Hello, everyone, and welcome to today's Lab Tools webinar. I'm Tiffany Garbett, Assistant Science Editor for The Scientist, and I'll be moderating our discussion. Today, our speaker, Dr. Abby Olsen, will discuss how an innovative Drosophila model helps explore the genetic contribution of glia to Parkinson's disease pathogenesis. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the speaker will address these during the Q&A session following the presentation. To ask a question, simply click on the Q&A tab and type your query into the question box located on the right side of your screen. We will try to address as many of these questions as we can during our Q&A session. The webinar will be archived on the Scientist website, and we will send you the link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsor, BioLegend. BioLegend offers a wide-ranging portfolio of tools and solutions for Parkinson's disease research, including antibodies, immunoassays, recombinant proteins, magnetic cell separation, and single-cell proteogenomics. BioLegend reagents were built in collaboration with preeminent leaders in the field, including the Michael J. Fox Foundation. BioLegend is committed to partnering with researchers to provide new and comprehensive solutions for studying neurological disorders. Our sponsor has provided us with some helpful resources related to today's webinar, and we have posted these in our handout section located on the right side of your screen. You can access and download these documents at any time during the webinar. And with that, let me introduce our speaker, Dr. Abby Olsen. Dr. Olsen is an associate neurologist at Brigham and Women's Hospital and Brigham and Women's Faulkner Hospital, and an instructor in neurology at Harvard Medical School. She graduated from Carleton College and completed her MD PhD at the University of Pennsylvania, followed by residency and fellowship training at Mass General Brigham. Dr. Olsen received an NINDS K08 Award and a Department of Defense Parkinson's Disease Early Investigator Award. Her research relies on a combination of Drosophila and human genetics to identify novel glial genes that contribute to the pathogenesis of Parkinson's disease and other alpha-synucleinopathies, with the long-term goal of developing glial-based therapeutics for alpha-synucleinopathies. She authored 18 publications and was recently selected as an American Neurological Association Emerging Scholar. In the clinic, she cares for patients with Parkinson's disease and multiple system atrophy. Dr. Olsen, let's just make sure that your slides are up and running and that all looks well. Perfect. Dr. Olsen, please take it away.
Okay, great. Thank you. I'd like to thank the Scientist as well as BioLegend for sponsoring the talk. And I'm delighted to be able to tell you today about my research on the role of glia in Parkinson's disease, as well as other related alpha-synucleinopathies. Just a couple of disclosures. My funding is from NINDS and the Department of Defense, and one antibody that was used in this talk was provided as a kind gift from BioLegend, the sponsor of the talk. Here's a brief outline of what I'm going to talk about. I'll start by giving you an introduction to alpha-synucleinopathies for people that aren't familiar. Then I'm going to tell you about our novel Drosophila model for understanding glial neuronal interactions in these diseases. And specifically, I'm going to give you 3 examples of how this model can be used to understand neurodegenerative disease. First, studying alpha-synuclein itself in glia. Second, moving beyond alpha-synuclein and looking at the role of other known Parkinson's disease genes in glia. And then finally, at the end, in a bit of a teaser, I'll show you some unpublished data about how we can use this model to identify novel glial genes that contribute to Parkinson's disease pathogenesis. So beginning with the introduction, alpha-synucleinopathies are neurodegenerative diseases. They're fairly devastating diseases, and although we have symptomatic therapy available, there are no disease-modifying therapies for any of these diseases. They're all defined pathologically by inclusions of alpha-synuclein protein that has misfolded. This causes death of dopaminergic neurons as well as many other types of neurons and results in motor impairment, autonomic dysfunction, cognitive symptoms, and many other symptoms. There's 3 specific diseases that fall under the broad category of alpha-synucleinopathies. Those are Parkinson's disease, the most common one, dementia with Lewy bodies, and a rare disease called multiple system atrophy. And all of these diseases, as I said, are defined by inclusions composed of alpha-synuclein. But what many people don't realize is that these inclusions are present in both neurons as well as glial cells to varying extents depending on the disease. So in Parkinson's and dementia with Lewy bodies, the inclusions are predominantly neuronal. These are the classic round Lewy bodies, as shown here on the bottom right. But there are also inclusions in astrocytes to a lesser extent, and that's shown in the top right. In contrast, in multiple system atrophy, the predominant inclusions are actually glial. They're found in oligodendrocytes, and these are termed glial cytoplasmic inclusions and are really the defining feature of the disease pathologically. Additionally, there are also some inclusions in neurons in multiple system atrophy. So this has been known for a long time that these inclusions can be in both neurons and glia, but the role of glial alpha-synuclein has really not been focused on. And in fact, the role of glia more broadly in alpha-synucleinopathies is not really well established, as much of the focus of research has been on the role of neurons. It's my opinion that glia have been overlooked as sources of potential therapeutic targets for these diseases, which, as I said, have no disease-modifying therapy. So the goal of my research is really to try to understand what glia are doing in these diseases in order to develop in the long term, disease-modifying therapies that are based in glia. So that's the end of the introduction. Now I'm going to tell you about our fly model for how we can understand glial neuronal interactions in these diseases in vivo. First, I want to take a moment to just tell you about some general advantages of Drosophila as a model, compared in particular to rodent models. So they have a short lifespan and generation time. You can do a whole experiment in a matter of days. They obviously have much, much smaller and simpler brains than humans, but they have surprisingly conserved functions, circuitry, and analogous regions. The genome in Drosophila is actually even more conserved compared to the human genome, with about 75% of disease-related genes having a fly ortholog. And there are numerous genetic tools available for working in the fly. Finally, flies have complex motor movements, as well as many important non-motor symptoms that are relevant to Parkinson's disease and other synucleinopathies. And you'll hear about some of these in my talk today. One question that I commonly get is, well, do Drosophila even have glia? And the answer is yes, they do. Without going into a great amount of detail, they have several kinds of glia. And what's important to know for purposes of this talk is simply that many of the functions of mammalian glia are conserved down to the fly, including protection of axons, involvement in the innate immune system, formation of a blood-brain barrier, etc. So that brings me to our specific model system, and understanding this slide is quite important for the entire talk, so I'm going to walk you through it fairly slowly. So if you start on the left in this cartoon, this is my depiction of a neuron here, and the way the model works is we use a two-part expression system with a pan-neuronal enhancer in order to drive expression of human wild-type alpha-synuclein in all neurons. This was published a couple of years ago. These flies develop Lewy body-like inclusions, so formation of alpha-synuclein inclusions. They have motor impairment and they have neurodegeneration, really recapitulating the key hallmarks of alpha-synucleinopathies. If we then look at the right of the cartoon, we can use a similar two-part expression system in glia and a pan-glial enhancer in order to drive the expression of any gene of interest in glia. And this is great because it's a very flexible system for studying neuron-glial interactions in vivo. So I'll show you a few different examples of how to use this system, which you can use to drive either overexpression of a transgene of interest or a short hairpin RNA to knock down any gene that you'd like. So now this will be the bulk of the talk. I'm going to show you some published work for how I use this model to understand the role of glial alpha-synuclein. And as I said in the introduction, this glial alpha-synuclein is particularly relevant to the disease multiple system atrophy. And so I just wanted to take a step back to tell you a little bit more about this rather rare disease. So this is much less common than Parkinson's disease, but unfortunately quite a bit more devastating. As you can see from this nice review in the New England Journal of Medicine, multiple system atrophy is a disease that really affects many different systems in the body. body. And unfortunately, it progresses fairly quickly. So from the time of diagnosis to the time of death is an average of 8 years. Shown another way, this MRI really illustrates how quickly the disease progresses. This is from a patient of my own with this disease, and her MRI in 2016 at the time of diagnosis is shown here. For those of you that are not accustomed to looking at MRIs, I'll just tell you that already this is a little bit normal, but even if you are abnormal rather, and even if you don't look at a lot of MRIs, I hope you can appreciate the difference here just 2 years later. So she's had really marked atrophy of the cerebellum, the pons, and the middle cerebellar peduncles, with now way too much space here in the back of the brain from loss of neurons. So just to drive home the importance of this, there was a recent meeting of the Global Multiple System Atrophy Research Roadmap Group, and among many recommendations they came up with was really a need for better preclinical models in order to hopefully make some progress in generating therapies for this disease. So that brings me back to my model diagram. And so, as I said, the way that this model works is we can express any gene of interest in glia. So in this first set of data, what I'm doing is expressing alpha-synuclein itself in glia. So all of the data that I'm going to show you will have human wild-type alpha-synuclein either expressed in glia, in neurons, in both cell types, or neither, allowing us to understand how alpha-synuclein acts in different cell types. So first, I developed a novel locomotion assay in order to examine behavior in these flies. So to give you a sense of what this assay looks like, these are control flies here, and you can see that they are happily moving around just fine. These are flies that have alpha-synuclein in both neurons and glia, and what I hope you can appreciate is they are moving quite a bit less. This is graphed over time here, and you can see that with age, even control flies, shown in black, have some decline in this assay as they get older. Flies with glial alpha-synuclein have a steeper decline, flies with neuronal alpha-synuclein have a steeper still decline, and then flies with alpha-synuclein in both neurons and glia have a very steep decline. In addition to difficulty moving, I wanted to assess some component of the autonomic nervous system in these flies. The autonomic nervous system is really heavily impacted in Parkinson's disease and even more so in multiple system atrophy. So I developed actually a way of testing constipation in the fly, believe it or not. This involves putting flies on blue-colored food for 24 hours. And then transferring them to regular food. And what you can see here is at the time of transfer to regular food, they all have blue abdomens from having eaten the blue food. And control flies that are normal and don't have alpha-synuclein pass this blue dye fairly quickly, whereas flies with an increasing alpha-synuclein burden are unable to do that, presumably due to constipation. That's quantified over here. Then moving beyond simply a phenotypic analysis to look at actual pathology, glial alpha-synuclein actually causes neurodegeneration as well. So we can see that at the top with vacuole formation. Or if I look at total neuron counts, you can see there's a stepwise decline here with increasing alpha-synuclein burden. And then perhaps more interestingly, if I look at dopaminergic neurons by measuring tyrosine hydroxylase staining, here again you can see that glial alpha-synuclein has effect. And in fact, it's a more marked effect. So if you compare total neuron loss here, which is a relatively modest effect with glial alpha-synuclein, and compare that to loss of dopaminergic neurons, you can see a much greater relative change. Similarly, when alpha-synuclein is in both neurons and glia, there's a really, really marked loss of dopaminergic neurons. And so this suggests that dopaminergic neurons are somehow sort of uniquely sensitive to the presence of glial alpha-synuclein. If we look at alpha-synuclein aggregation in these cells, and here is where I'm using an antibody that was given to the lab from BioLegend that proved to be quite helpful for these studies. What we can see is that alpha-synuclein aggregates in the cells in which I've expressed it. So when it's expressed in glia, it aggregates in glia and not in neurons. When it's expressed in neurons, it aggregates in neurons and not in glia. And when it's expressed in both cell types, you see aggregates in both cell types. What's perhaps more interesting than that is, again, looking here at dopaminergic neurons stained with tyrosine hydroxylase staining in red and alpha-synuclein in green. What you can see is that in the both condition, where alpha-synuclein is in both neurons and glia, there's actually even more alpha-synuclein inclusions than when it's only expressed in neurons, again suggesting non-cell autonomous effects of glial alpha-synuclein on worsening alpha-synuclein in dopaminergic neurons. To get a little bit mechanistic, I then did bulk RNA-seq from whole brain here, looking at transcriptome changes from glial or neuronal alpha-synuclein. And here these results were quite surprising, to me at least. If you start by looking at this Venn diagram of genes that were altered with either glial or neuronal alpha-synuclein, what you can see is that there's really very, very little overlap. So it's a quite different set of genes that were upregulated with glial alpha-synuclein as opposed to those that were changed with neuronal alpha-synuclein. Additionally, if we look at the direction of change over here on the left, with glial alpha-synuclein, nearly all of the genes were upregulated, whereas with neuronal alpha-synuclein, nearly all of the genes are downregulated. So it's not only a different set of genes, but also the direction of change that's different. In the condition with alpha-synuclein in both neurons and glia, this looks very similar to neuronal alpha-synuclein alone, actually. And I think that the reason for that is likely because glia represent only about 10% of cells in the Drosophila brain. And this is bulk RNA-seq, so I think that the signal is mostly swamped out by the neuronal signal. If we look at specific pathways that are altered in the RNA-seq, with glial alpha-synuclein, proteolysis genes were changed a lot, upregulated, as well as cell surface receptor signaling genes. And these are interesting. These proteolysis genes are a bunch of secreted proteases, so this may represent a compensatory response to the presence of alpha-synuclein. These cell surface receptor signaling genes are also interesting. They include many tetraspanins, and these are genes whose human orthologs are specifically enriched in oligodendrocytes, which, if you remember from the beginning, are really the cells implicated in multiple system atrophy. So this tells us that our fly model of alpha-synuclein in glia is likely recapitulating some of the pathology of the human disease. In contrast, the genes that were downregulated in the presence of neuronal alpha-synuclein were largely related to lipid metabolism, both very generally and also specifically. There were many genes that were downregulated that are related to fatty acid metabolism. And this is interesting because it actually really nicely synergizes with some other work coming out of a couple of groups at Brigham and Women's, identifying fatty acid metabolism in particular as a new target for Parkinson's disease. Finally, just to drive this home, this is a graph here with some of the genes we found in the RNA-seq shown here at the top, divided into pathways shown here on the y-axis. And these are genes whose mouse or human orthologs have previously already been identified either in rodent models of multiple system atrophy or in human genetic studies. And so again, that just suggests that our fly model is able to pick up on genetic changes that are already known to be important to the human disease, as well as many novel genetic changes. So to just summarize this part of the talk, this is a novel model for understanding glial alpha-synuclein that we hope will shed some light on this devastating disease, multiple system atrophy. I've shown that glial alpha-synuclein impairs locomotion, it causes constipation and induces neurodegeneration, and it also augments all of these phenotypes when combined with neuronal alpha-synuclein. Interestingly, dopaminergic neurons appear to be disproportionately sensitive to the presence of glial alpha-synuclein. And in fact, glial alpha-synuclein induces aggregation of neuronal alpha-synuclein in a non-cell autonomous fashion. Finally, glial versus neuronal alpha-synuclein induce distinct transcriptional programs, suggesting that the cell context of alpha-synuclein expression really matters for its effects. And in both cases, they induce transcriptional changes that seem to be relevant to human alpha-synucleinopathies. So that data was all published last year. I'm now going to move on and show you a couple of other exciting uses of this model that involve unpublished data, and this is all work in progress, so I hope you'll keep that in mind. So I'm moving away now from multiple system atrophy and back to Parkinson's disease. Unlike multiple system atrophy, Parkinson's disease is a quite common disease. It affects 1% of people over the age of 65, which is 10 million people worldwide. Again, its pathology consists of misfolded alpha-synuclein that forms these Lewy bodies in neurons, as well as to a lesser extent inclusions in astrocytes. This causes death of dopaminergic neurons in the substantia nigra and really progressive motor impairment. And here, for those of you who are not clinicians, I thought it might be interesting to show you just a patient video. This is one of my patients, so that you can see the effects of Parkinson's disease firsthand. This patient has early Parkinson's disease, for what it's worth, so her symptoms will get worse over time. But let me play the video here.
Okay, go ahead and tap 10 times with your right hand.
So here I'm asking her to tap on the right, and this is normal.
And now with your left.
And on the left, you can see it starts out normal and then becomes slow and small.
Okay, good. And now with your right hand, I want you to open and close 10 times.
So similarly, this is normal on the right.
And then now with your left.
And again, starts normal and gets very slow and progressively smaller. So that's called motor decrement or bradykinesia, and it's one of the hallmarks of Parkinson's disease. That symptom will get worse for her in the coming years, again, because we have no disease-modifying therapy. So bringing it back to the science now, there are many reasons to think that glia are important in Parkinson's disease. One of those reasons is genetic. Unlike multiple system atrophy, we know quite a lot now about the genetics of Parkinson's disease. So human genome-wide association studies have identified at this point nearly 90 risk loci for Parkinson's. These studies, in my opinion, have high face validity in that some of the loci identified include the alpha-synuclein locus, LRRK2, and GBA, and these are all genes that are known to cause familial variants of Parkinson's disease due to point mutations, suggesting that they really are, the GWAS are really finding, real pathologic genes here. That said, there are many issues with getting from GWAS to actually understanding disease pathogenesis. For one, most GWAS studies have focused on genes that influence risk of development of the disease. Those genes might be entirely different than those that drive actual progression of the disease once it's established. For another reason, GWAS identifies SNPs, and the assumption is that the SNP is controlling expression of nearby genes, but in some cases that assumption may be incorrect, and so we need EQTL analysis to really understand how these SNPs are involved in gene expression. And then finally, and of most interest to me, GWAS is really agnostic to the cell type of interest, meaning that the signal in a GWAS might come from neuronal expression of that gene, glial expression, or even expression from cells outside the brain. And if you look over here on the right, what I've done here is take all of the current GWAS candidates here on the y-axis, and I have use publicly available RNA-seq data from Ben Barris's group to look at their expression across brain cell populations. And what you can see is that actually only a minority of the genes are really predominantly expressed in neurons. Many more are expressed predominantly in some type of glia, or they're just sort of ubiquitously expressed across the population of cells. So for all of these reasons, we really need mechanistic studies to be able to move from GWAS to an understanding of disease pathogenesis. So here I'm again returning to the fly model system, and here I'll show you a second way of using it. So again, we have human wild-type alpha-synuclein expressed in all neurons, and this time, rather than expressing alpha-synuclein in glia, I will express a short hairpin RNA in order to knock down Parkinson's GWAS candidate genes in glia to see if they have a role there. So we did a small screen using this approach back in 2015. We knocked down the majority of Parkinson's GWAS candidate genes that were known at that time. It's a much smaller list for what it's worth than the current list of 90. We found 6 candidates that were promising in the initial screen, and then ultimately pursued 4 of them after validating them. Those 4 are listed here, and I've listed their human ortholog in parentheses. First showing you that all of these glial modifiers, they're all enhancers, so they all make the phenotype worse. So in each case, when the gene is knocked down in glia, there's more impaired locomotion compared to just the presence of alpha-synuclein in neurons alone. As one small technical note for one of these 4 genes, OX, there's a lot of toxicity when it's expressed using a pan-glial driver. So for this one alone, I'm expressing it in astrocyte-like glia only. All of the others are expressed in all glia, and that's why OX is going to be shown on a different graph for all of the upcoming slides. In addition to enhancing the locomotion deficit, knockdown of all these genes in glia also enhances neurodegeneration as measured by both total neuron counts and dopaminergic neurons. And now, interestingly, this is where the results start to diverge. So I've shown you that all 4 of these genes enhance the locomotor deficit and enhance neurodegeneration. If I then start looking at some more specific phenotypes that are part of Parkinson's disease, we start to see a divergence in some of these glial modifiers. So here I'm showing you some bioenergetic data. This is using a seahorse machine. And what we can see is that 3 out of the 4 genes actually impair the bioenergetic status of the brain, but one of them, OX, does not. If we then look at alpha-synuclein aggregation, there's further divergence. So 2 of the glial modifiers, LRC and RIC, increase alpha-synuclein aggregates, whereas VPS13 had no effect. And OX, interestingly, actually decreases alpha-synuclein aggregates compared to its appropriate genetic control. To dive a little bit more into the mechanism of what might be behind change in alpha-synuclein aggregates, I looked at autophagy markers, and here I'm looking at P62, a marker of autophagosomes, and with glial LRC knockdown, this is increased, whereas the other genes really didn't have any effect on it, and OX knockdown actually decreased autophagosome markers, similar to the decrease in alpha-synuclein aggregation. I see similar results with a different autophagy marker, LC3, again suggesting that glial LRC knockdown is impairing flux through the autophagy pathway. I then wondered whether these autophagosomes that are increased in glia themselves or increased in neurons, and I believe they're increased in neurons. So shown here at low magnification is staining for glia with the marker repo, and LC3, one of the autophagosome markers. And in alpha-synuclein flies, there are somewhat modest amount of these autophagosome markers. Some of them are sort of nearby glia, but not a ton, and not obviously co-localizing with the glial marker. When we have glial LRC knockdown that really increases the number of these green puncta, they still don't particularly seem to be associated with glia. Shown at higher magnification, again, we see occasional ones that are sort of next to the glia, but really the bulk of them, including in cells that have a whole lot of impaired autophagy, are sort of more adjacent to the glia. So I believe that this is the non-cell autonomous effect, and these markers are actually being increased in neurons in response to glial LRC knockdown. Likewise, if I look at co-localization with simply alpha-synuclein as a marker for the neurons, and LC3, the autophagosome marker, you can see that largely these inclusions appear to be in cells that express alpha-synuclein, i.e. neurons. So to summarize this part of the talk, again, these are somewhat preliminary unpublished data, but what I've shown you here is that some GWAS candidates exert at least some of their effects in glia. So this suggests that manipulating genes in glia causes non-cell autonomous injury and loss of neurons. Beyond that, though, it's a little bit more complicated. So not only can we manipulate genes in glia and cause neuronal death, what I've shown you here is that different GWAS modifiers actually affect different aspects of Parkinson's pathogenesis when we look at specific pathways. So that really argues against a non-specific glial injury type of model or a reactive gliosis model in which just hurting glia hurts the neurons in a non-specific way. So what I think is really going on actually is that in fact, specific glial genes control specific parts of the pathogenesis. So while in all cases we get here to dopaminergic neuron death, there might be many routes by which we can get there. And in fact, it's even more complicated than that because some of these processes are affected by only one modifier, whereas some of them span several modifiers, making it really challenging actually to come up with a linear model for how you get from A to B. So this is very much work still in progress, but I want to end the talk by actually telling you about a third application of this model, which is the one that I'm really the most excited about. So I've already shown you that we can use this model to understand glial alpha-synuclein. Then I've shown you that we can use it to start to understand the mechanism of other Parkinson's disease genes. Now I want to move beyond known Parkinson's disease genes and show you how we can use it to identify novel genes. So yet again, returning to the model slide, here, once again, we have alpha-synuclein expressed in all neurons. And this time in glia, I'm doing an RNAi-based screen to knock down the entire kinome in Drosophila. This screen is ongoing. So far, I have knocked down 238 kinases out of about 360 that Drosophila have. Kinases are highly conserved between fly and human for what it's worth. I'm using my locomotion assay as a readout for this screen. And what you can see is that about half the genes have no effect when knocked down in glia. 12% are lethal even when knocked down only in glia. 9% are toxic, meaning that they actually made locomotion worse in control flies in addition to alpha-synuclein expressing flies. 21% are enhancers, so these would be like the GWAS genes that I've shown you that make things worse. And then finally, the set that I am the most excited about is the small piece of the pie that are suppressors. So 6% of the genes are suppressors, meaning that when knocked down in glia, they actually rescue the alpha-synuclein phenotype in neurons. So these are the genes that are potentially good therapeutic targets. So I've deliberately obscured these results because this is very new data. But what you can see here is a string network analysis of the results I have so far. And what you can see is that there are starting to be clusters observed in this network. So this tells us something about specific signaling pathways that when perturbed in glia can modify neuronal alpha-synuclein toxicity. And I'm going to tell you a little bit more about the biggest cluster so far, which I'm quite interested in. And this is purine metabolism. So if you're going to mention metabolism in a talk, I think you're obligated to show one of these horrific diagrams just to make the point that purine metabolism, like all metabolism, is very complicated. But I will try to take you through a couple of key ideas here. So first, a brief refresher on purines. If you're like me and haven't thought about what on earth is a purine in a long time, chemically they're defined by a pyrimidine ring that's fused to an imidazole ring. And here are several important, biologically important purines here to give you some examples. They're quite an important class of molecules in general. So just showing you the derivatives of adenine. Adenine, of course, is the backbone of DNA. Adenosine is an essential signaling molecule that has widespread effects in the body. And then, of course, we have AMP, ADP, and ATP, which are critical for energy metabolism. Beyond their general biologic importance, purines are also of known importance in Parkinson's disease in a couple of ways. So first, the downstream metabolites of adenosine, in particular urate, the end product of purine metabolism in humans, urate is known to have antioxidant effects and is thought to be protective in Parkinson's disease based on epidemiologic studies. But the story is fairly complicated because there are also some purine antagonists, for example, caffeine, which is an adenosine A2A receptor antagonist, that has also been shown to be protective in Parkinson's disease. So there's a lot of need for further understanding of these pathways and how different specific purines affect the disease. Returning to the big scary diagram, we have reasons to believe in our fly model of Parkinson's that purine metabolism may be altered. So based on my own RNA-seq data that I presented to you at the beginning, as well as proteomics data that's from a postdoc in the lab, Suvar Sarkar, this is currently under review for publication, many genes involved in purine metabolism have altered expression in the fly alpha-synuclein model. And these genes fall into sort of two categories. First, at the top, there are genes that are involved in de novo purine synthesis, so the genes that code for enzymes that make purines. And then second, down here, there are several enzymes that alter the composition of purines. So they're involved from switching from one purine to another. And these are the ones that I'm really going to be focusing on. So just to give you a sense of what the results of the screen look like, I've identified quite a few purine genes. And so rather than focus on any one, I want to give you sort of a broad idea of what they look like. So returning to these videos here, again, we have control flies, and you can see them nicely moving around. These are flies with neuronal alpha-synuclein, and they move less. If I show you an example of a glial enhancer, so this is a gene when knocked down in glia, and you can see that basically the flies pretty much don't move at all. In contrast, if we have a glial suppressor, so a gene, again, only knocked down in glia, you can rescue the entire phenotype and the flies are moving great. Similarly, if we look beyond behavior, but at actual neurodegeneration, the results are analogous. So with neuronal alpha-synuclein, there's a loss of neurons compared to control flies. If we add a suppressor RNAi, you can largely rescue this, so it looks similar to control. Whereas if we add an enhancer RNAi, the flies are really just quite devastated, as you can see here. So this is basically my final slide with some data, and I just am going to try to summarize the ongoing results from my studies in purine metabolism. Essentially what I think is happening is that these various genes that I've knocked down that are either enhancers or suppressors are all converging on glial adenosine. I've shown in the alpha-synuclein flies that they have decreased adenosine in the brain in general. And I think by increasing the production of adenosine from glia, that is rescuing much of the pathology. And the reason I think that is that knockdown of the genes that would be predicted to lead to increased glial adenosine, those are all the suppressors, whereas knockdown of genes that would be predicted to decrease glial adenosine are all the enhancers. So this is my current working hypothesis. Just to summarize this last part of the talk, I've shown you a third use of this Drosophila model, which is doing a forward genetic screen, knocking down the kinome. This has led to identification of purine metabolism as an important pathway in pathogenesis, and has allowed me to generate the hypothesis that increased glial adenosine is neuroprotective. So I hope this shows you how we can get from unbiased screens towards real hypothesis-driven research. There are many reasons to think that glial adenosine might be protective. Astrocytes are the major source of adenosine in the brain. There's quite a lot of evidence for signaling between neurons and astrocytes using adenosine and ATP. And also there's a lot of evidence from the stroke and seizure literature that adenosine can protect against excitotoxicity. So that may be what's going on here, but it's still early days for this project. Okay, I'll end with a global summary of the talk, which is that no disease-modifying therapies exist for alpha-synucleinopathies. So there's really a critical need for new basic models to try to understand the pathogenesis of these diseases in order to ultimately develop therapies. My opinion is that the role of glia in these diseases has really been overlooked, and so we may be missing glial genes that could be therapeutic targets. To try to remedy that problem, we've created a novel Drosophila model for understanding glial neuronal interactions in alpha-synucleinopathies. And I've shown you today 3 different ways that this model can be used. First, to study alpha-synuclein in glia. Second, to study other Parkinson's genes beyond alpha-synuclein in glia. And then finally, and most interestingly to me, how we can use the model to actually identify novel Parkinson's disease glial genes. This is a tool that can be used both to identify genes and understand their mechanism, and it can allow us to prioritize potential therapeutic candidates for investigation in mammalian systems. And that brings me to, I believe my second to last slide, and that's just to leave you with the big picture of my lab. Everything I've told you today is about Drosophila, which I believe is a wonderful tool, but it is only one of many tools. So much of my focus these days is on taking these genes identified in the fly and moving them into glial neuronal co-culture systems, as well as rodent models of Parkinson's disease. With the idea in the long run, again, of doing human translational studies and eventually clinical trials to try to come up with new therapies for these diseases. I just want to end with some acknowledgements. In particular, I'd like to thank both BioLegend and the scientist, again, for the opportunity to talk to you today. I want to thank my technician, Sabrina Clemens, as well as my whole mentorship team who are becoming collaborators now. They are truly wonderful. And then finally, my funding sources again. And with that, I'm happy to take questions.
Wonderful. Thank you for that presentation. As a reminder to our audience, you may submit your questions at any time to Dr. Olsen using the Q&A tab. Our audience has already submitted several questions, so let's get to them. Dr. Olsen, after you identify important glial genes in the fly, how do you prioritize which ones to study in mammals?
Yeah, so there's several things that I take into consideration there. So a lot of my focus is on whether or not genes might be good therapeutic targets. And so one of the things I take into consideration is whether there are any FDA-approved drugs like already targeting a gene of interest that I've identified in a screen, for example. Another is how the genes fall into pathways. So I showed you string network analysis, for example, and I'm sort of more interested in the genes that are clustering together, suggesting that there's a whole cellular pathway of importance as opposed to sort of a one-off isolated gene. I also consider how good of an ortholog it is between the fly and human. In the kinome, all of the genes are conserved actually because the kinome is so important, but still some of them are more conserved than others. So I prioritize ones where there's high conservation between the fly and the human in the hopes that, you know, that would be a good target in humans as well.
Thank you. Do you think that particular subsets of glia are more responsible for driving the pathologies described here?
Yeah, good question. I think that they may all be important, but in different ways. So, for example, the purine metabolism things that I've identified in the screen, the best evidence from the literature would suggest that that's probably taking place in astrocytes. And also, for what it's worth, among the subtypes of glia, astrocytes are probably the most conserved between fly and human. So it may well be that much of what I'm finding is particularly relevant in astrocytes. That said, I think all of the types of glia likely have a role in these diseases. Certainly there's great work coming out of like Beth Stevens's lab, for example, showing how microglia prune synapses, and that's quite important in neurodegeneration. I told you about multiple system atrophy, where the primary pathology is actually in oligodendrocytes, and there's problems with myelination. So I think that all glia may be relevant, but much of my work is probably particularly relevant for astrocytes.
Great, thank you. Our next question is, with regards to the LRRK2, the knockdown of that particular gene, it seems to occur in opposite directions, where it disrupts autophagy in the glia, but increases it in neurons. How do you reconcile this sort of like opposite directional effect that's happening with that knockdown?
Yeah, that's a good question. So I'm still trying to understand the mechanism of what's happening there. Just to take one step back, that gene LRRK2 is a very interesting gene in general. So in work that I'm not able to show you, it seems like it's probably very tightly regulated because it worsens the phenotype when knocked down in glia, when knocked down in neurons, or when overexpressed in glia or neurons. And there are actually mutations of that gene that are thought to be maybe gain of function genes, or mutations rather, that actually cause human Parkinson's disease. So it seems like there's maybe a real critical threshold for LRRK2 expression where any aberration is bad. It's also an extremely large protein. It is both a kinase and a GTPase and has roles as a scaffolding protein. So some of it might have to do with its different interaction partners, of which there are many, as well as different functions of the kinase activity versus the GTPase activity. So that's an area of really active research in the lab, is trying to understand the many functions of LRRK. And in terms of the non-cell autonomous effect of, how does knocking it down in glia alter autophagy in neurons? That is a very good question. We're trying to figure out if there is a secreted factor somehow that is altered by LRRK knockdown in glia that is then acting on the neurons. But that research is very much ongoing, so I don't know the full answer.
Fascinating. For emerging evidence suggests that alpha-synuclein and other proteins responsible for neurodegenerative diseases can act like prions. Can this model be used to study spread of these proteins?
Yes, I think so. So that's something that I have not fully focused on, but it's a question that I get a lot because this is a very exciting area of Parkinson's disease research in general. So there's mounting evidence from rodent models, for example, that alpha-synuclein can act in sort of a prion-like fashion, where if you inject aggregates or preformed fibrils into the mouse brain, it can function as a seed to cause aggregation of endogenous mouse alpha-synuclein, for example. It's not yet clear the extent to which that might be relevant to the human disease, but that, again, is an area of active research. And what's nice about our fly model is that because we can control the expression of alpha-synuclein using any tissue-specific driver, we can precisely control the cells in which alpha-synuclein starts. And then if we see it wind up in other cells, then it has to have gotten there by some sort of spread. And so I think this is an interesting model for potentially examining that question.
Great, thank you. It may be too early to say, but can adenosine slow the progression of the loss of dopamine cells?
Yeah, good question. So again, that's really active ongoing research. I have shown for some of the suppressors that I think upregulated adenosine, that they can rescue the neuron counts. So yes, they can slow the progression. But this is actually a subject of an R01 that I'm currently writing, where I'm planning on testing this pathway in human iPSC-derived glia and neuron co-cultures and in a mouse model of Parkinson's disease. And so if that ends up working, that will be really valuable evidence that this pathway is important in mammals as well as in the fly.
Wow, cool. What happens when the shRNA is expressed from an inducible promoter that is expressed after the alpha-synuclein has already caused locomotion defects? Is there still a protective effect by the knockdown of genes in purine metabolism, you think?
Oh, that's a great question. So I haven't done that yet, so I can't answer it, but I would love to know that. And I think that's a really important question because you're absolutely right. That would be much more analogous to the situation with human disease, where, of course, we don't know who will get Parkinson's in advance, so we can't simply give them extra adenosine or something ahead of time, but rather we're trying to use these things once the disease is already established. So I think that's a very good question, but I have not actually tested it yet myself.
Okay. With regards to the MSA studies, why do you think there's this sort of cumulative effect that's happening between the neurons and the glia, and both having lost that gene, that we see this sort of double effect there?
Right. Yeah. So I think that's a great question as well. And the RNA-seq study is designed to get at that a little bit, although there's still much mechanistic work to be done. So some of it, sometimes people argue simply that perturbing anything in glia is going to be bad for neurons because glia are the support cells of the brain. So they provide quite a lot of metabolic support to neurons. They protect the axons, etc. But I do think it's more specific than that being the only issue because, for example, if we overexpress other genes in glia, such as, for example, ataxin-3, which causes a spinocerebellar ataxia, a different neurodegenerative disease, it has no effect on neuronal alpha-synuclein toxicity. Likewise, there are genes that we can knock down that have no effect on neuronal alpha-synuclein toxicity. So it's not simply that perturbing, any old thing in glia has an effect. I do think there's some degree of specificity to it. And I guess the best hint that I have so far would be from the RNA-seq data, where I see all these changes in these tetraspanins, the cell surface receptor signaling genes. Those are genes that are involved in the human in oligodendrocyte maturation. And so I think somehow alpha-synuclein in glia is really injuring the glia themselves, and that likely changes signaling between the glia and the neurons and exacerbates neuronal injury. But there's still much work to be done to try to understand what specific molecules are involved.
Great, thank you. How did you decide which candidates to test for the shRNA studies?
Yeah, good question. So my plan in the long term is actually to do a whole genome screen, or rather a conserved genome screen, meaning that I would like to knock down every gene in the fly genome that has a human ortholog. But that takes years, even in the fly. So as sort of a starting point for just sort of proof of concept that the model is useful, that's why I started with Parkinson's disease GWAS genes, because those are genes that we have a strong reason to think that they're important in the disease. And then I moved on from there to the kinome, mostly because kinases are so very druggable. And at the end of the day, I am a physician scientist and I see these patients and really do want to develop new therapies. So to me, it makes sense to start with genes for which there might already be an FDA approved drug that could potentially be repurposed as a therapeutic for Parkinson's disease. So that's why I've started there. I'm expanding from the kinome into the whole druggable proteome, as it were. So that's about 600 genes that are already targets of FDA approved drugs. And then from there, I plan to eventually do the whole genome.
Wow. For those shRNA studies, did you look at any reactions in vivo, or were they all in vitro-based studies?
Yeah, so this is all in vivo in the fly brain, basically. So the locomotion assay is in the whole fly, as is the constipation assay. And then at 10 days of life, we fix the flies and we can do pathologic studies on brain slices. So I hope that answers the question.
Sort of going back to the big picture, does alpha-synuclein have any normal function in the body, so that this is something that is normal, that has perhaps gone awry? Why did you choose this avenue?
Yeah, that's also a good question. It has been surprisingly hard to answer. So a vast number of people have tried to figure out exactly what alpha-synuclein usually does. It is clear that it resides mostly in synapses in a physiologic state, and it probably plays some role in corralling synaptic vesicle proteins. That said, alpha-synuclein knockout mice, for example, have actually a very mild phenotype. They're basically fine. So it's surprisingly tolerable to lose alpha-synuclein. And that has made it a little bit of a challenge to understand what it does. There's also evidence from my mentor, Dr. Malfini, that it may regulate the actin cytoskeleton and participate in some very basic biology, and that part of what goes awry with it might have to do to excessive actin cytoskeleton stabilization. There's even evidence for it having a role in DNA repair. So people have proposed all kinds of different physiologic roles of alpha-synuclein, but it's been quite hard to figure out. And that part of the mystery, actually, of why people even get Parkinson's disease is what starts this process and goes wrong. It's still really unknown.
Great, thank you. And now we have time for just one more question. How does the fly help you move from identifying genes in a screen to really understanding their mechanism?
Right, so we can take advantage of lots of genetic tools that are available in the fly, only some of which I've been able to show you here. But by using, tissue-specific promoters, we can tightly regulate expression of genes. For example, I have shown you things that are expressed in all neurons or all glia, but I can also express things just in dopaminergic neurons or just in astrocyte like glia using various promoters, and that can allow us to figure out which cells specifically are important for various phenotypes. There's also quite a lot of genetic reporter tools available for the fly. So for example, one thing that I'm working on right now is looking at calcium reporters or cyclic AMP reporters to try to understand some of the downstream signaling events that are maybe altered by altering these purine metabolism pathways. And so using all those various genetic tools available in Drosophila, we can do quite a lot in vivo that would just be completely impractical to do in a rodent model.
Fascinating work. Unfortunately, that's all we have time for today. If you have any further questions, please consider reaching out to the speaker directly. Her email address is shown on the screen. As a reminder, the webinar will be archived on the scientist website, and you will receive an email notifying you when the on-demand webinar is available. I would like to thank everyone who took the time to join us today, and particularly those of you who shared your questions and comments. On behalf of the scientists, I'd also like to thank our speaker, Dr. Abby Olsen, as well as our sponsor, BioLegend. Thank you, everyone, and have a great rest of your day. Goodbye.